Hello everyone, I'm Lucinda McKenzie, or at least that's my scientific name, and this is Oscar the fire ant. I didn't know if you could see Oscar very well, maybe that will help. We're here to help you get ready for the Wolbachia Rodeo. So you got to be rodeo ready because it is a STEMulating competition. Yeah, you can tell this is a very high budget video. Well, it's a competition in that students are really competing against themselves to try to get the best results possible. In order to have a great STEM project, it must be real and it must be relevant. Mosquitoes and fire ants are real problems in our community. Mosquitoes are vectors that can carry all sorts of diseases that can cause West Nile virus, chikungunya, yellow fever, types of encephalitis, and even cause heartworms in dogs. Fire ants hurt when they sting and destroy cropland, and I should probably make a video of just how many ant beds have cropped up in my yard just in the past month. So, we're testing local insects to see if they're infected with a ubiquitous bacteria called Wolbachia, which is currently being studied by scientists to see if it's as a possible biological control. So how can your students help? Using biotechnology, they will extract DNA from the insects, and if Wolbachia is in the insect, that DNA will be extracted also. And they're going to use PCR to determine if our local insects are infected with Wolbachia. So, how do you get started as far as getting ready for a rodeo? Usually at the rodeo, there will be a rodeo baggie prepared for you, or at least partly prepared. If you have the protocol for the Wolbachia rodeo, I'm going over some of the items on here that will allow you to get rodeo ready. One of the first things that you will see, you need one sealed 1 millimeter micropipette tip. So a P1000 pipetter would use these 1 mil tips because 1 mil and 1,000 microliters are the same thing. So they need to be sealed. You might have to get your students to check. Oh my goodness, I'm not rodeo ready. Hold on a second. Okie dokie, oh my goodness. Oscar, you didn't even tell me. Okay. So, to seal your tip, you might have a nice friend like Oscar, who is not very nice and not reminding me, or you might have a Bic lighter, which stands for Bee Insect Collectors, because students, you need to be collecting these, playing a very active role, and as you know, when it comes to insects, you can't stop the insects, but maybe we can work at controlling some of the insects. Okay, so back to the macerator. Flick your Bic. It doesn't take but just a fraction of a second. Just put it in the edge of that flame. Oh, I have on prescription glasses. Students, make sure that you put on your safety glasses. When you're doing this, you don't want any plastic flying off and popping you in your cornea. I wish we had some safety glasses for Oscar, but I don't right now. So, check it. If it's sealed, you're done with that. Go to the next one and transform that microchip into a macerator. So, flick your Bic or have a candle ready. Just the outer edge. It doesn't take long. You just want to seal it. Now, students have worked on different designs, whether it's more of a sharp pointed tip or a rounded edge. You get to be an engineer, a macerator engineer. You are designing macerators. You do that to all 5 if they are not already sealed. So you should have a lighter in your baggie. Okay, now the next thing, toothpicks. Toothpicks are mainly used to manipulate the insects. They're not going to be used for macerating. So after your macerators have cooled, if you want to just put that in there until you're ready to use it at the rodeo, that will be fine. Let's see, 2 groups of colored tubes. Now at the rodeo we usually do 5 insects, 3 that you bring, and then we have 2 controls. Usually it's a Nasonia negative and a Nasonia positive wasp, and so that's 5 samples. you should have 5 tubes, and I try to do the different groups and schools, different color tubes. It just helps when they're sharing microcentrifuges and different things together. The 1.5 mil tube is necessary for the initial maceration. Now the second group of 5 tubes can be smaller ones if you have them. If not, just go with the 1.5 again. Because the smaller ones, the 0.65 microliter tubes, are only going to be used to store the purified DNA, and you're only collecting 50 microliters. So if you don't have them though, that's fine, no problem. Go ahead with the larger ones. Okay, 1 sheet of paper towel, which in the next video I'll show you how to label that. A lot of times the insects have been stored in 91% isopropyl alcohol, and you need to remove that alcohol so the DNA does not precipitate during the reaction that we will be running. So just 1 sheet of paper towel. You can see it's cut into a little strip. It doesn't have to be a whole sheet. You also need, distilled water. I usually go ahead and aliquot out the distilled water. The protocol says 100 microliters per insect. That's because we're working with small insects. If it's large, you might need a larger amount. Go ahead and aliquot that out. You can fill the tube up. It doesn't really matter unless water's price goes up between now and then. And then Instagene. For some reason, I like just putting in a green tube. I think of green with Instagene. I don't know why. Instagene, you need 200 microliters per insect. We're doing 5 insects. So 5 times 2, there should be 1,000 microliters in here. It is up to the student team to check to make sure there's 1,000 microliters in here, and there's a little line that says 1.0. It's a good thing that you know your metric conversions, and you know that 1 milliliter is the same as 1,000, what, Oscar? Microliters. That's right. Thank you, Oscar. Okay. So you check that, and here's the unique thing about Instagene. It settles out. So you need to shake it. You know that song, Shake It Like a Polaroid Picture? Oscar, you remember that song? Once you shake it, you should not be able to see through it. This is so important because later on when you start running the protocol, you have to shake this every time before you withdraw any Instagene. Otherwise, you may not get the pellets that are necessary for chelating the magnesium ion. Okay, so I do not usually hand out the primers at this time because they're not needed until you're ready to do PCR. So before the rodeo ever starts, I go ahead to save time because you're doing this rodeo in one day. I go ahead and aliquot out distilled water. I just get it from Walmart. And the Instagene. The Instagene you can buy by itself, or you can, if you use Biorad, some of their PCR kits come with Instagene, and I usually had some left over that I could sort of incorporate into the rodeo to save on expense. Now, if you don't have any of that, this is designed to be a simulation. So students, stop listening, put your fingers in your ear. Teachers, for simulated Instagene, I just get some baking powder, add it to water because baking powder will settle out. It gives the effect, but when they get to the rodeo, then they can work with the real thing. Okay, tell them to get their fingers out of their ears. Okay, welcome back, students. Hope you enjoyed that nice little break. Okay, so the other things that are listed, base pair, ladder, loading die, PCR beads, you do not need it to get ready. This is all you should have in your baggie. So again, if you're doing 3 insects that you bring in, and usually at the rodeo we do in alphabetical order, ant, Solenopsis invicta, also called the red imported fire ant, a flea, because they are vectors also. I did not mention him. I hope he didn't get his feelings hurt, fleas that are listening. Sorry. And mosquitoes. We test 3 types of mosquitoes, but it just depends on the luck of the draw which one you get. If you bring in your own, then you know what it's going to be. We work with Anopheles species. We work with Aedes albopictus, and then we also have different species of Culex. So it may be your lucky day. I don't know which one you'll get until you get to the rodeo. So, ant, flea, mosquito, and then the rodeo usually provides your two controls, the Nasonia negative and the Nasonia positive. So all of this is listed at the top of the protocol, and those are the things that should be in your rodeo baggie. But again, the macerators may not be sealed, and usually if there's a big lighter sitting at your station, that's a clue to you to let you know, check those. If they're not sealed, you get some time being an engineer and designing your own macerators, which I think is great. So anyway, I'm Lucinda McKenzie, Oscar the Fire Ant. We hope to see you at the rodeo. Thank you.